We were discussing this Sark homology domains, really, right? There are many Sark homology domains. These domains are very important in signaling because they are the actual signaling intermediates. They can interact with the receptors once they are getting phosphorylated. One of the reasons I just told you, the phosphate group is negatively charged. In this case, the Oncogene residue in the SH2 domain is positively charged, and this makes an ideal situation to interact because there is one side positive charge, another side negative charge, so they will be able to easily interact. The other thing that you have to remember is that these modules of or the domains are usually about 100 amino acids. In this case, they bind to specific phosphotyrosine containing residues or peptide motifs. I have already told you the dissociation constants are about 50 to 500 nanomolar in this case. And another important thing is you may have to remember this structure. The SH2 domains usually contain a central antiparallel beta sheet. This is a beta sheet flanked by 2 alpha helices. So this is what you may have to remember. SH2 domains, as I already mentioned, they bind to phosphotyrosine. You may also remember that the receptor tyrosine kinases in their cytoplasmic domain contain several tyrosines. So this makes it very ideal for these groups of the domains to bind with the phosphotyrosine containing areas. So each one can bind to say if there are 10 molecules, they can bind to 10 different residues, right? So that should be possible. This is one important concept to understand, that this can to some extent diversify the signals to different molecules. These domains usually act like adapters. You know what is an adapter that we use in the house? We just plug it in and then we can connect 2-3 plugs in that. So it is a plugging device where multi-plugs can be inserted into one plug. So immediately you understand that one becomes many. So that way it multiplies the signals. So why we are talking about that here is because similar to SH2 domains, there are also SH3 domains, SH4 domains, pH domains, several domains are there. And each one will interact with a certain set of proteins. So not only that, one protein molecule may have more than one domain. Same molecule may have SH2 domain, SH3 domain, and SH4 domain, and pH domain, and things like that. So this gives an advantage for these interacting molecules to diversify the signal and also to kind of amplify the signals. So the examples given here are the PDGF receptor, which is a platelet derived growth factor receptor, which is a receptor tyrosine kinase. So it contains 5 tyrosine residues. Interestingly, the tyrosine kinase domain that is usually present in the cytosol is split into 2 domains. So it is called a split tyrosine kinase domain. So you can see that here, that's shown in green here. So before the split, you see, after the split, the first subunit, you see that it binds to PI3 kinase, GTPase activating protein. And then just below that, after the second domain, you have 2 tyrosine residues. They bind to phospholipase C gamma enzyme. It also contains SH2 domains and SH3 domains, as you can see here. So this is a very important concept to kind of understand. Then what is called, we are now talking about different domains, the so-called phosphotyrosine binding domain. Again, it is about 100 to 150 residue containing domain, and they commonly bind this particular motif. This is what asparagine, proline, X means any amino acid, and tyrosine. So this motif must be there for it to bind. The PTP domain will bind only to, in other words, it will bind to this particular motif where there is tyrosine also. Remember that. The phosphotyrosine binding domain of the docking proteins, other docking proteins include what is called SHIC, SHC, and IRS, insulin receptor substrate 1, require ligand phosphorylation on the tyrosine residue for binding. So these residues, they require phosphorylated tyrosine residue. The PTP domains such as so-called X11, BAB, FE65 and NUM apparently recognize the so-called motif NPXPY or related peptide motifs, but they are not dependent on ligand phosphorylation. Why this sentence is given here is because don't think it is always dependent on ligand phosphorylation. Usually it is dependent on ligand phosphorylation. So sometimes they are not dependent on ligand phosphorylation. This is another important domain called SH3 domain. The importance of this is that it will bind to proline rich peptides with the minimal consensus shown there. Proline, XX means another any amino acid, PRO again proline. So two proline residues are interrupted with two other residues. So two amino acid residues are flanked by proline. You can also say that way with the same. So each proline is usually preceded by an aliphatic residue. Each of these aliphatic proline pairs bind with a hydrophobic pocket on the SH3 domain. Again, the ligand binding site consists of a hydrophobic patch that contains a cluster of aromatic residues surrounded by charged and variable loops. As usual, the binding affinity and specificity can be increased by interactions involving loops on the SH3 domain. I hope there is a, did we show that SH3 domain? No. Yeah, okay, doesn't matter. This is another one called pH domain, nothing to do with our pH. This is plextrin homology domain. We call it pH domain. This is also again about 120 amino acids. They also have very high affinity to specific phosphoinositides. I hope you know what is inositol. Cyclohexane with the cyclohexane is what? 6 carbon. In each of them you will have a OH, that is inositol. So you can say hexahydroxycyclohexane if you want to in a chemistry language. So that is inositol. And the positions of the phosphate bonds are shown there, like in the 4th position and 5th position if you have phosphorylation, it is called 4,5 inositol 4,5 biphosphate or bisphosphate. If the phosphate groups are there in 3, 4, 5, then it is called phosphatidyl inositol 3, 4, 5 triphosphate. The diphosphate can be 4, 5 or 3, 4. That's what we call it as phosphatidyl inositol PI, either 3, 4, 5 or 3, 4 or 4, 5. These are the residues which can be phosphorylated. Accordingly, you can have bisphosphates or triphosphates. Binding to these phosphoinositides may allow pH domains to respond to lipid messengers. Thereby they can, for example, this kind of signal can be important in relocation to membranes. From cytosol they may go to membrane. That signal may be given. Now we have given several such examples. As I already told you, one molecule may contain more than one domain or even have repeats. For example, look at this, it is called GRB2. This has two SH3 domains and one SH2 domain. Similarly, you have, let us take some of the kinases. For example, SRC is important. It is having one SH2 domain and one SH3 domain. And then let us look at phosphatase, it's called SHP2, SHP2. You can see that it contains two SH2 domains. You can look at that here. Similarly, in the RAS signaling, you have what is called RAS gap proteins. They have pH domains, SH3 domain, and SH2 domains also. This tells you that one molecule may have more than one domain or even 2-3 domains, same type, of the same type. SH2, two SH2 domains can be there, two SH3 domains can be there, or one SH3 and two SH2 domains, like that. So this is one idea that we must have, because that tells us how the signal can be diversified and amplified. For amplification, we have some more examples, I will come to that. Then some of the proteins involved in ubiquitination, like here Sibyl is shown. So they have SH2 domain and a ring domain and so on. Then cytoskeletal regulation also involves such as tensine, chimerin and all that. And this is another important molecule, the last one, it's not last but not the least, PLC gamma. And before that also the so-called SOCS, suppressor of cytokine signaling. That is the full form of that. It contains also SH2 domain. Similarly, this PLC gammas, we have already talked about it. pH domain, SH2 domain and SH3 domain, and even 3 pH domains are there. So you can understand that. Now, the interaction between different signaling molecules, the so-called signaling intermediates, are all mediated by these modular proteins. As I told you, adapter proteins. So they will be in between. So they connect. Their job is to connect two different proteins with the receptor. It's not that it always it has to be connected with the receptor. Receptor may be connected to one domain, and that domain may be connecting with several other proteins. So it acts like a circuit. So that is very interesting. You can look at it here. For example, let us take one receptor, some receptor, doesn't matter. So the receptor gets phosphorylated and immediately it interacts with this particular protein, let us say signaling protein 1. Now this signaling protein 1 contains a phosphotyrosine binding domain, so it immediately connects it to signaling protein 2. Not only that, the signaling protein 1, which is connected to the receptor, also connects with another ligand or messenger, in this case through its pH domain. For example, see the connections. What is very important is that one molecule, the so-called signaling protein 1, gets connected to, for example, inositol phospholipid and then to the receptor itself. So it is at least connecting to two. Not only that, it is also connecting to another signaling protein 2. So if you take signaling protein 1 itself, it connects to 3 different molecules. What are the 3 different molecules? Signaling protein 2, inositol phospholipid and the receptor. So this is a very important example. Then the adapter proteins, again some of the adapter proteins, they can connect to another signaling protein, for example. then the signal is relayed downstream. So this is how signaling takes place. This is a very important thing. Now the signaling efficiency can be further enhanced by what are called scaffolding proteins. For example, in this case, this one shown here is a scaffolding protein. It interacts with not only the receptor but three different protein kinases. Three different protein kinases. The advantage of protein kinases, it comes in the next slide. What happens is they can create what is called a phosphorylation cascade. What is it? You can see here. This is a very important slide. Let us take a signaling molecule. It binds to a receptor and it gets phosphorylated. And now it phosphorylates, the receptor phosphorylates an inactive protein kinase, which gets activated by this phosphorylation. Now it becomes, it activates another protein kinase. That is, in other words, the first kinase gets activated by phosphorylation, and the kinase 1 now phosphorylates another kinase. By this phosphorylation, it activates a kinase. All the kinases here shown in this example are activated by phosphorylation. So one kinase activates another kinase, and that activates another kinase, that activates another kinase. This is called phosphorylation cascade. This is very important concept to understand. Finally, they may result in the translocation of a transcription factor into the nucleus, and that may result in binding of a specific sequence in the DNA, which results in the transcription, and that may mediate a particular cellular response. Can be proliferation, growth inhibition, or apoptosis, or whatever. That may be a particular cellular response. So these are the sequence of events. It's very important to understand that many kinases are activated by phosphorylation. The same thing is also true that they can be inactivated by dephosphorylation. So both kinases, what is the enzyme that dephosphorylates? Phosphatases. So this is the important thing in signal transduction. But it's only, as I said, it is only usual. There are exceptions. You cannot say that all the time phosphorylation activates. I gave you an example, RB. It gets inactivated by phosphorylation. So you cannot take it verbatim. So it depends on the situation. So that is shown here, and some explanation is also given. You can look at it. So the book I am referring to describe this kind of cell signaling and all that is by Molecular Biology of the Cell by Bruce Alberts. Now, in cancer, naturally, the signal transduction cascades are undergoing some aberrations. There will be some abnormalities. Either it can be over functioning or it may be suppressed. As I told you in cancer biology, generally we say that the signaling pathways can be oncogenic signaling pathways or tumor suppressor signaling pathways. Both can be there. Naturally, growth factors and their receptors play an important role. So I have given some examples here. You can look at it. EGF, for example. EGF is what? Epidermal growth factor, and there is one TGF alpha, which is transforming growth factor alpha. PDGF is platelet derived growth factor. This is vasculoendothelial growth factor, VEGF. So these are some examples of growth factors that essentially we are giving these examples to those which bind to receptor tyrosine kinases. These growth factors are also called mitogens because of their ability to induce cell proliferation. Those which can induce cell proliferation are generally termed as mitogens. Growth factors are essential for cell survival. Cells in culture generally requires serum. The purpose of adding serum is that the serum contains a variety of growth factors. So these growth factors, I told you the examples given are those that bind to receptor tyrosine kinases, which are usually present on the surface, cell surface. Now, how the receptor function becomes deregulated? That is the question. What happens is sometimes the regulatory domains, maybe see the extracellular domain, may be deleted in some cases. In some cases, there can be an amplification, overexpression of the receptor, overexpression or gene amplification of the receptor may also be an abnormality. And this results in constitutive dimerization. If you remember, I told you what is constitutive activation, or in this case, dimerization. That is spontaneous, without a ligand even. There can be constitutive dimerization. If there is a loss of regulatory domain, you don't need a ligand. The receptor will get automatically firing, start firing. So that is the important thing. Sometimes due to gene fusion and all that. Here the example given are again receptor tyrosine kinases, which typically contain an extracellular domain, transmembrane domain, and intracellular domain, and a cytoplasmic. So generally they require a ligand for firing the signal, but if some mutations or overexpression or deletion of the regulatory domain happens, then there can be ligand independent firing of the signal. Firing means signal is getting transduced at a more than normal rate. That's why we call that as, in this case, ligand independent firing. That's what happens generally. That means what? There will be more and more proliferation. Of course, receptors can become deregulated also in the case of cancer cells by their autocrine signaling. I told you they will produce more and more growth factors, and they have their own receptors, the very same cells that produce them. So thereby there can be autocrine signaling. SRC is a tyrosine kinase. Don't ever get confused with SRC. SRC is not a receptor tyrosine kinase. It is a non-receptor tyrosine kinase. It's a tyrosine kinase. It is present in the cytosol, for example. It's not a receptor tyrosine kinase. Whenever we say RTK or receptor tyrosine kinase, we mean the one that is attached to the membrane, which contains an extracellular domain, transmembrane domain, and a cytoplasmic domain. Whereas SRC is not like that. But the importance of that is like it is the first cellular oncoprotein to be studied. We discussed how SRC was discovered and all that. Generally, although we talk a lot about tyrosine kinases, serine threonine kinases are also important because more than 99% of the phosphoamino acids in the normal cells are phosphothreonine and phosphoserine. That doesn't mean phosphotyrosine is not important. It's only 0.05 to 0.1% of the total phosphoamino acids in the cell that are phosphorylated at tyrosine residues. But they are also very, very important. EGFR is the famous example for a tyrosine, receptor tyrosine kinase. As I told you, the extracellular domain is also called ectodomain, transmembrane domain, cytoplasmic domain. The cytoplasmic domain is there, and another, what is the, okay, there is a SRC homology domain also in the cytoplasmic region. That is what is shown here. The very important thing is that when the growth factor molecules bind to the receptor, there is transphosphorylation. Actually, if both are EGF receptors, you call it as autophosphorylation, but it is actually one kinase phosphorylating the other. So there must be two molecules involved. It is in fact a transphosphorylation because one kinase, one receptor phosphorylates the other. Since they happen to be the same EGFR, for example, in this case, both are EGFR, that is called autophosphorylation. Autophosphorylation, generally textbook definition is self-phosphorylation. But students are tempted to think that one receptor is there, it will bend and phosphorylate itself. No, it's not the case. Actually, two receptors are there, they will phosphorylate each other. This is an important concept to understand. Here some examples of receptor tyrosine kinases are given. It's very important. Even the orphan receptor that I told you, HER2 new, is also a receptor tyrosine kinase because it belongs to the EGF receptor family. So, another interesting thing is on the outside, for example, in EGF receptor, there are what are known as cysteine-rich domains that contains a lot of cysteine residues. Another interesting feature is displayed by insulin receptor, IGF-1, insulin-like growth factor 1 receptor it's called. See, it has actually two residues and they are linked by disulfide bridges. So it is an alpha chain and beta chain. Actually, you can see that they are like dimers in nature itself. So they have this kind of structural features. And some receptors like the so-called NGF receptor, which is nerve growth factor receptor, PDGF receptor, platelet derived growth factor receptor, and colony stimulating factor receptor, macrophage colony stimulating factor receptor. That's what is called MCSF. FGF is fibroblast growth factor receptor. All of them, interestingly, and this VEGF receptor, vasculoendothelial growth factor receptor, all of them contain on their extracellular domain, they have what is called immunoglobulin-like domains. And some other receptor tyrosine kinases, such as F receptor, they contain fibronectin type 3-like domains. So, just you have to understand that there are different receptor tyrosine kinases and they contain different kinds of domains on the extracellular portion. Integrins also play an important role. As you know, the normal cells require an attachment. They require some kind of a support. This is often called anchorage dependence, or sometimes they call substrate dependent also. The cancer cells, they don't have this property. They don't need anchorage. So they are said to be anchorage independent. That means they can grow in suspension. They don't need a support. The moment cells come into contact with each other, they stop growing, the normal cells. That is called contact inhibition. Whereas cancer cells, they don't have this contact inhibition. They grow one over the top of another. This is known as loss of contact inhibition. Similarly, anchorage dependence also. Normal cells are anchorage dependent. The cancer cells are anchorage independent. In the absence of attachment, that is, if you don't have the support, then the normal cells undergo some kind of a cell death and that is called anoikis. The way by which cells understand this or sense this is through cell surface receptors called integrins. So integrins are very important. So if integrins are there, then the cells will understand whether there is a substrate or not, and if it is not there, they are going to commit some kind of cell death. That's called anoikis. Don't confuse with apoptosis, it's different. Signals transduced by integrins, therefore, affect cell shape and motility. So this is an important concept also understand. I generally followed the biology of cancer by Weinberg. Another important concept that you may have to keep in mind, that is the aberrations. I will discuss some of the most important aberrations that occur in cancer in the next class. But before that, I just want to say that when we say aberrations, it can occur at any step of the signal transduction process. What are the different steps? The defects can be at the ligand level, or the defects can be at the receptor level, or alternatively, the defects can be mutations or something, deletions, all those kind of things could happen at the signaling intermediate level. Not only that, finally, some of the transcription factors or the transcription complex, many members are there, they can also get some kind of aberration. And finally, the product that is formed, which is responsible for a particular function, may also become defective due to something or other, structural changes. So basically, all these things can happen either alone or together. So this has to be kept in mind to get an overall picture. Many cancers that happens. So if we know if the cancer cells are addicted to a particular aberration, then it is often called oncogene addiction. That means this kind of aberration gives a growth advantage to the cells so that they continue to grow. That produces something like a chain reaction. So if you stop this, you can stop the chain reactions. But you should also understand cancer is something where many genes are deregulated. So you generally, when you are attempting to give some therapy or treatment, you are attempting to set right many genes, not one gene, multiple genes. But one exception is if the cells are addicted to one gene, like this oncogene addiction, then if you set this right, you may be able to treat. So this is practically happening. It's not something theoretical. Practically also. One example I already gave, but later on when we discuss the molecularly targeted therapy, we will discuss more examples. The example that I already told you is with reference to HER2 new oncogene, which is overexpressed in 20 to 30% of breast and ovarian cancers, where if you come out with an antibody to that antibody will neutralize this and thereby will reduce the effect of this overexpression of HER2 new, and that is a treatment modality. Herceptin, the antibody is called, is one of the most important antibodies used in cancer therapy. Although there are many other antibodies which are now in the clinic. Okay, so I think we'll stop here today.